Hey guys, Dr. K here. Time again for another capsule. This time we'll be talking about placebo use in phase 1 studies.
Hey, sorry I'm late. What did I miss?
No, actually, I was just about to start.
I was thinking, if I look just like you, that means I'm a placebo.
Well, not really, because placebo may look the same, but there's no active ingredients inside.
Well, you could say that I am an inactive ingredient in your capsule, so by definition, I would be a placebo.
Okay, well, anyways, let's get started. So in today's capsule, we'll be talking about placebos used for IPs that are administered through IV infusion pumps, syringe drivers. I'll give you different blinding tricks that can be used. We'll also discuss a bit about over-encapsulation, suspension placebos, and taste masking. So a question we often get from clients is, I have an API that needs to be administered. We haven't studied it yet, but we don't have a placebo. We're not necessarily ready to invest in a CDMO to have it developed. Is there a way that we can start our first trials without it? And the answer is yes, because if your API needs to be administered through IV infusion, we'll just use a saline solution. But then the question is, like, it has to be double blind. So how can you ensure that it won't be unblinded if the API changes a little bit, either the color of the solution or if it's a bit cloudy and things like that? Well, the simplest way to do it, and what we do regularly, is just to cover all of the parts with an amber bag. So as you can see here on our little videos, I have an example here where we have an IV bag with a nice bright blue API inside. So the amber bag covers it well. We'll close it at the bottom. So then the nurse administering the product won't be able to see through it. So she won't be able to determine between a clear bag and the one with the API. The rest of the extension set will also be covered with an amber bag to hide the coloration in the extension set. And for syringe drivers, it's the same the same idea that we're going to use. We will use these amber shields that will cover over it. So in the example here, we see that the bright yellow API looks the same now as the clear saline solution after the shield is being put. Same thing if we have to add that extension set, it's going to be also covered. If your product is not for IV, it's for oral use. If it's just a powder, you can have it added to a capsule. So in the pharmacy manual will be detailed the number of milligrams that's added, it's weighed, and then we can add a filler. The placebo in this case will use the same capsule, the same color, and just fill it with an inactive ingredient instead of the API. If your API is like little tablets, but you need to do a blinded study, we can add those little tablets to a capsule, add a filler, and the placebo will be only the filler. Same idea if you have a double-blind study where you have an active control. We'll put the active control over encapsulated filler, and the same thing with the drug, and the same thing for the placebo. If the API is for oral use but ends up being a suspension, well, then there's different things that come into account. Because first of all, a suspension, the API is floating, so placebo will have nothing that's floating. So same idea. We have to add an inactive ingredient that's going to mimic the precipitation that's being done by the API. So we can use, let's say, cornstarch. But then the other problem is that they won't precipitate necessarily at the same rate. So then if one precipitates faster than the other, it could give you an idea of which one is the API versus the placebo. So the important thing is trying to keep it in suspension as long as we can. So it's important to mix it appropriately. So stir plate, homogenizers, and then when your doses are prepared, you can use a rocking shaker to have the IP syringes moving to stop it from precipitating as fast as it can. And very important on the IP admin forms to have that your nurse has to shake before they're removed, because we would put them in, like, say, amber bags, so to hide it if it started precipitating. So to shake the bag with the syringes inside prior to use. And then when they take it out, the appearance will still be the same before administering it to the subject. Then if the API has a nasty taste, well, one subject will only get one product. But if they're talking to each other, which may happen, one's going to say, oh, it has really, really strong bad taste, and the other person will say, no, it doesn't taste like anything. Then between themselves, and then if the clinical staff hears, they could determine which one received the active API and which ones got placebo. So in these cases, when the taste is very pronounced, we can add like taste masking to it. So by adding flavoring like sucralose, fructose to the formulation of the placebo and the test, what's going to help mask that difference so they won't notice it as much. When you're buying those products, it's important to take a look at your sucralose, that it's a USP grade. If that's not available, you can always go for NF grade, but nothing that is used as food additives. So that pretty much covers my little presentation for today. I hope you enjoyed it. So please follow me, and I'll talk to you next time.